name is Tony Futterman. I'm the senior author on this paper. I've been working on sphingolipids for about 35 years. And in this paper, we attempt to introduce a new concept that lipid bilayers are actually finely tuned molecular assemblies. The concept of fine-tuning comes from physics and from cosmology, and it's to do with the fact that the physical constants in the universe are perfectly fine-tuned to allow life to develop in the universe. In this paper, we go on to look at a number of parameters of the lipid bilayer, which supports our concept that lipid bilayers are actually fine-tuned.
Hello, my name is Tamir Dingjan. I'm the first author on this paper. The question raised in this review is, are lipid bilayers fine-tuned for life? How much can they change and remain compatible for life? To answer this question, we looked at the composition, asymmetry, and interactions present in the eukaryote plasma membrane. First, we consider the composition of the bilayer. How much can the mole percent composition of a lipid bilayer change before the function of the bilayer is affected? We point out in the paper that there are way more lipids than anyone ever imagined. We are now aware of at least 45,000 unique lipid structures, and up to 1 million different lipids may exist in nature. At the present, in typical mass spectrometry studies of lipid bilayers, we can identify up to 400 different lipids. How tolerant are cellular bilayers to these changes in their lipid composition? The answer is it depends on the function. Different individuals have about 84% of the lipids in their platelets in common, so clearly at the whole cell level, quite large changes could be accommodated by living cells. But more compelling evidence of fine-tuning comes from studies in liposomes, where adding between 1 to 5 mole% of ceramide lipids can cause the formation of detergent-resistant membranes or ceramide-enriched domains. So altering lipid composition of the cellular bilayers by small amounts can change their biophysical properties, and this supports the idea that their composition is fine-tuned for life. Next, we considered the asymmetry of lipid bilayers, the distribution of lipid species between the upper and lower leaflet of the bilayer. You can see here the breakdown of lipid species in human red blood cell plasma membranes by the number of double bonds in their acyl tails. The leaflets have a clearly different distribution between the upper and lower leaflets. How much can this asymmetry change in living cells? It turns out not very much. Consider the case of phosphatidylserine. This lipid is typically held entirely on the inner leaflet of the plasma membrane and is only exposed during apoptosis. Exposure of as little as 3 mole% of this lipid on the outside leaflet is an eat me flag that triggers phagocytosis. We suggest that with such sensitivities, the bilayer asymmetry is a finely tuned phenomenon. Finally, we looked at the interactions between lipids and membrane proteins. An example is the specific interaction of sphingomyelin C18 with the copy protein P24, in which only the 18 carbon length sphingoid base is bound. A difference in chain length of 2 carbon atoms is sufficient to almost completely disrupt the binding interaction. We also discussed the agreement between the asymmetry of leaflet packing and the asymmetry of transmembrane domains in proteins. Transmembrane domains span both leaflets of the bilayer and are, on average, thinner in the outer half and thicker in the inner half. This corresponds to the difference in lipid packing between the tighter outer leaflet and looser inner leaflets of the cell plasma membrane. Changing the width of the inner and outer halves of a transmembrane peptide can change the degree of plasma membrane localization, so we consider this a very fine-tuned interaction. So, to summarize, this paper presents lipid bilayers as a finely tuned assembly of molecules which show multiple fine-tuned phenomena. Far from being a freely diffusing collection of randomly selected amphiphilic molecules, we find that the lipid bilayer has a surprising degree of functional complexity. We are very excited to see how experimental techniques can detect even finer tolerances in lipid bilayers in the future. That's a quick overview of our paper. If you found that interesting, please head to BioEssays, where you can find the whole review.